And just taking a step back, ALK testing in general, most people doing IHC, NGS, FISH, what is kind of the standard at the institutions?
So ours has been now sort of a standard. Everybody gets sort of a reflex testing, and we still do the FISH because that's been the established sort of mechanism. But I think there's good data for IHC at this point.
IHC is an approved approach, and we use IHC because it's faster.
Right.
Is it a part of your next-gen profile? Have you worked that in, or you don't have that?
But in terms of immediate results, I mean, I think that the IHC assays are much faster.
See, that's the point. You know, if you send a next-gen sequence off, you know, we do the Oncomine at Yale now. It can take many weeks to come back.
Yeah.
But, you know, for us, we really need 3 or 4 things that we need to know before we can treat.
Exactly.
So you're right, the IHC, I think, is, and then you can confirm it with FISH if you need to.
As Suresh mentioned, this field has moved incredibly at a rapid pace. The discovery of ALK in 2007 with crizotinib being developed and proved in 2011 is one of the quickest times ever between the discovery of a mutation and the development of a drug. We have, as mentioned, multiple now ALK-directed therapies that are approved. One of those is Borghaei. We have data with Borghaei in the crizotinib refractory space. Tom, you want to talk to us about the Borghaei data? Because we're trying to learn how to keep up with all these new drugs.
It's hard, and it's a dynamic landscape, as you know. So Borghaei is a very potent ALK inhibitor that has activity against many of the ALK resistance mutations. And this was a randomized phase 2 in the post-crizotinib patient population that looked at 2 doses, 90 milligrams daily, or 90 milligrams for 7 days, followed by 180 milligrams daily. And what it found is the response rate is around 50% with both of those schedules. And the PFS in the 90 milligram was around 9 months and about 12 months in the 90 followed by 180. And it was fairly well tolerated. We saw significant responses in the CNS. This drug has a unique toxicity in the early onset pulmonary symptoms, and they can occur in about 5% of patients, generally in the first week. And that's why you have to do the 90 milligrams for a week and then dose escalate up to 180 milligrams as part of the toxicity management. I personally think the drug has a lot of promise, but I think the post-crizotinib setting is not really the patient population that we're seeing right now. Right. I mean, because now the question is, what's the post-alectinib therapy, and how are we treating those patients?
I had one patient who did get crizotinib first, then alectinib, and then got brigatinib, and actually had a pretty remarkable response to the brigatinib that lasted about 7 months.
Yeah. So. And we need more data post-alectinib. I think it's nice to have post-crizotinib data. We have that with brigatinib. We've got that with suritinib. We have that with alectinib, and they seem to compare favorably. Although the PFS with brigatinib post-crizotinib is a little bit longer than the other 2 drugs, but I think it's tough to know what to do with this drug. Has anybody used it in the treatment-naive setting? It's not approved in that way, but has anybody used it?
I haven't used that treatment-naive, no.
No, there's
hard to do that with alectinib.
Right.
Right. And there's a phase 3 trial of the treatment-naive, and so I think that's good. And I think Ram brought up the critical point. When we do these trials, it's very important to get tissue because many times the people who have the ALK mutations are going to be more susceptible to more potent ALK inhibition. But if they have a bypass track, we're probably not going to get a lot of bang for our buck with hitting the ALK pathway a little bit harder.
Exactly.
How do we interrogate post-alectinib? Is it through plasma? Is it through rebiopsies? I think there's some ongoing data. Do people routinely do this in their practice?
Plasma
well, a couple of times that I've done the plasma, I've been a little bit of a setback because if a patient doesn't have huge tumor burden and they have minor progression, you are unlikely to get a lot of information out of serum-based assays. I mean, you can correct me if I'm wrong, but that's been my general experience. So I think in the patients like that, if you really are thinking about switching, you might be obligated to do a tumor biopsy, try to figure out what the mechanism of resistance is.
Yeah.
I don't know what everybody else's experience
In a
is.
clinical trial setting, that's good information to know.
Right.
And I think increasingly I'm testing it even outside of a clinical trial. I want to know if the patient has G1202 or not, because I might find a trial for that particular mutation. But for routine practice at this point, I would say the resistance mechanism perhaps doesn't change therapy options.
Right. Yeah.
So it's soon to come advance, I would say.
I think we need to mandate, I mean, those trials, of course, mandate tissue and plasma so we can understand how to direct these patients based on these mechanisms of resistance. The last drug that I'll talk about is lorlatinib, which is yet another ALK-directed therapy, which is quite potent. We've had some data interestingly presented at ASCO last year, 2017, and it's been updated, looking at lorlatinib in a highly pretreated group of patients. We had cohort 1, which was a group that was treatment naive, that was ALK therapy naive, and the response rate there was 90%. That was a small number of patients, 30 patients, but 27 out of 30 responded. Interestingly, though, the group cohort 4 and 5, who had either gotten 2 prior ALK-directed therapies or 3 prior ALK-directed therapies, with or without chemotherapy, and those roughly combined in that group 111 patients, and the response rate was north of 40%, which is pretty interesting to get that kind of response after that many ALK-directed therapies. Now, I don't know which therapies there were included there, but in a PFS of around 7 months. So again, I think we're trying to learn how to sequence these drugs. Lorlatinib certainly has activity against G1202R, probably the most potent activity against G1202R, so it may be a nice fit post-alectinib. Any experience with lorlatinib thus far in the group?
So I have used it as part of clinical trials with lorlatinib, and I found this to be a good drug. And the trial that you mentioned that I took home, the key number was for patients with G1202, the response rate was 58% with lorlatinib, which is incredible. Yeah. And I think this drug will fill an important unmet need for ALK-positive patients when it becomes eventually available in the clinical practice.
It's very specific. It's patients who have been on it have told me that the side effect profile is just very, very mild.
It's got an interesting side effect with the hyperlipidemia or that has to be followed. But other than that, I think that the drugs, triglycerides,
right. And then some CNS side effects. They're grade 1 or 2 or something.
But the rest is quite mild.
Yeah. So we'll just have to see. I think we have an embarrassment of riches in the ALK space, and we'll have to better understand how to sequence these drugs, and the jury's still out, I think, but we've certainly made significant advances in this space over the past 5 years.